Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on regulation of glomerular filtration. Now, before I start this lecture, I would like to show you a few numbers. See, we have already discussed about the glomerular filtration rate GFR, and in young adult male, this GFR, we know the GFR for young adult male is 125 ml per minute. So in men per minute 125 ml filtrate is produced, and in case of young adult female GFR is 105 ml per minute. So in woman 105 ml of filtrate is produced per minute. Now, if you calculate this GFR on daily basis, so the number will be the amount of glomerular filtration will be 180 liter per day in case of male, and in female it will be 150 liter glomerular filtrate will be produced per day in female. Okay, now we know in the filtrate there present the water, then the nutritive substances are there, electrolytes are there, waste material are also present. So we can expel only water and waste material, but the electrolytes and nutritive substances, they are vital, they are important for our body. That's why kidney reabsorbs 99% of the filtrate, and kidneys excrete only 1 to 2 liter of urine per day. Okay, so the question is how this glomerular filtration is controlled, how it is regulated, what are the mechanism. So see, regulation of glomerular filtration. See, GFR must be precisely controlled because it is a very sensitive process. Okay, glomerular filtration is very sensitive process, and so this process need to be precisely controlled. See, if GFR is very high, then what happen? Fluid will flow through the renal tubule. Large amount of glomerular filtrate pass to the entire renal tubule, and this fluid is flowing very rapidly. As the fluid is flowing in large amount and it is flowing very speedily, so the renal tubule, the different parts of the renal tubule, they will not get the proper time to reabsorb the nutritive substances and electrolytes and water. Okay, so see, and large amount of filtrate is passing through renal tubule, so that time, that means the large amount of urine is going to be produced. Okay, and if person is urinating the large volume of urine, that means the person is going to be suffer from dehydration. Okay, and also in person body electrolytes are going to depleted. Okay, so and vice versa, if gfr gfr is too low, now what happen in this case? So in this case, from renal tubule the glomerular filtrate fluid will be moving slowly. It flows sluggishly. So now the renal tubule is having enough time to reabsorb the substances. So reabsorption, see, waste materials, they need to be excreted out of body, but as the fluid is sluggishly flowing through the tubule, what happened? The renal tubule, different part of the renal tubule, they are going to reabsorb the waste material. So we need to eliminate the waste material through urine. But now what happened? These waste material are reabsorbed and they enter into the blood. Okay. So the condition that arise due to the increased proportion of waste material in the blood, that condition is known as azotemia. Okay. Now see, the only way to adjust GFR from moment to moment is to change the glomerular blood pressure. Okay, so it is required that the glomerular blood pressure changes time to time. And how this glomerular blood pressure is going to change moment to moment? So this is achieved by three homeostatic mechanism. One mechanism is renal autoregulation, second is sympathetic control, and the third is hormonal control. So we are discussing one by one all these three mechanism. Now first we are discussing renal auto regulation. Now, what is the meaning of renal auto regulation? Renal means kidney, okay. Auto means by its own, and regulation means control, okay. And here we are talking about the GFR, glomerular filtration rate. Now, renal auto regulation is the ability of the nephron. It is the nephron's ability to adjust their own blood flow. So nephrons are able to decide that how much blood is going to flow through them. Okay. Then nephrons are also having the ability to control this GFR. And for this, nephron do not require any external control. Okay, that means the control from nervous system they don't require, or the hormonal control are also not required by the nephrons. So the nephron by their own, they can control the blood flow. Okay, they can control the GFR. Now now it enables them to maintain. So as the nephrons are capable in maintaining the blood flow and GFR, this enables nephrons to maintain a relatively stable GFR. Okay, nephrons are maintaining GFR in spite of changes in arterial blood pressure. So in our body, if there is a change in blood pressure, even in that particular situation, the nephrons are able to control the GFR, glomerular filtration rate. Now, if the mean arterial pressure, in short, MAP, MAP risen up from 100 to 125 mmHg, and there were no renal autoregulation. So, in this case, what will happen? Urine output would increase from the normal 1 to 2 liter per day to more than 45 liter per day. So what is the meaning? See, in normal condition, we urinate generally 1 to 2 liter of urine per day. But in case in our body, the arterial blood pressure, if it goes up from 100 to 25 mmHg, in this case, if there is no renal autoregulation, okay, the nephrons are not regulating GFR. So that time, what will happen? So that time we will be urinating 45 liter urine per day. But we are not urinating 45 liter urine per day. So what is the mechanism? See, renal autoregulation is there. Okay, here urine output increases only few percent. Okay, even if the blood pressure of artery, mean arterial pressure, increases up to the level of, even if the MAP rises as high as 160 mmHg, in that situation also we are not going to urinate 45-liter urine per day. Okay, so there is a auto regulation mechanism is there. Okay, now how this renal autoregulation is helping us. Okay, so this renal autoregulation help us to ensure stable fluid and electrolyte balance in spite of the many circumstances that substantially alter one's blood pressure. Okay, so it is the renal auto-regulation mechanism in which, so in, let's say our body's blood pressure has increased. So in that circumstances also, this renal auto-regulation enable us to maintain the glomerular filtration, and we are in that condition also when the blood pressure is high. In that situation also we are urinating normally, the normal volume of urine we are urinating. Now, there are two mechanisms of autoregulation. One is myogenic mechanism, and second one is the tubulo-glomerular feedback mechanism. So one by one, we are discussing both this mechanism. Now, first we are discussing the myogenic mechanism. Now, this is the mechanism of stabilizing the GFR. Okay, so this mechanism stabilizes the GFR, it controls the GFR, and this mechanism is based on the tendency of smooth muscle to contract when stretched. Okay, once again, smooth muscle are having tendency to contract when they are stretched. See, when the arterial blood pressure rises, it stretches the afferent arteriole. Okay, and now the arteriole constrict and prevent blood flow in the glomerulus from changing very much. So what is the meaning of this thing is that, see, this is the this one is the afferent arteriole, this is the efferent arteriole, and here the tuft of capillary. Okay, this is the glomerulus they are showing. Now imagine a condition that the blood pressure has increased, that means large volume of blood is entering in the glomerulus. Now, as the blood pressure is high and large volume of blood is entering into the glomerulus, so this incoming blood through this apparent arteriole is going to apply a pressure on the apparent arterial wall, and in the apparent arterial wall the smooth muscle cells are present. Okay, so now as these cells, smooth muscle cell, are experiencing this pressure by the blood, so they are going to, this wall is going to stretch, and now the smooth muscle cell, they are going to contract, and when the smooth muscle cell in the wall they contract, so this, so the diameter of the afferent arteriole is going to reduce, and if the diameter is going to reduce, that means now the lesser volume of blood is going to be supplied to this glomerulus capillaries, and now less amount of blood is entering into glomerulus, that means the glomerular filtration rate is going to be normalized, it will be under control now. Okay, this is one way. Now second, see, Now, conversely, now opposite to this situation, in this situation we have discussed the blood pressure was high and large volume of blood was entering into glomerulus. Now, conversely, opposite to this situation, imagine when blood pressure falls. Now, in this situation, what will happen? The apparent arteriole, this one, is going to relax. Okay, so that means the smooth muscle which are present in the wall, they are also going to relax. Now as the blood pressure is low, arteriole is relaxing, smooth muscle are relaxing. So now what happened, this allows blood to flow more easily into the glomerulus. So now in this condition, blood is entering easily in the glomerulus. And now, in even if the blood pressure is low in this condition also, as the afferent arteriole afferent this afferent arteriole is relaxing and it is dilated, so a good amount of blood is entering into this glomerulus and they are producing the normal glomerular filtrate. Okay, so this is known as the myogenic mechanism. Now we are discussing the tubuloglomerular feedback mechanism. Now this is a mechanism by which glomerulus tuft of capillary network which is present inside the Bowman capsule. So this glomerulus is receiving feedback on the status of the downstream tubular fluid. Okay, and adjust the filtration to regulate its composition. So here it is the glomerulus which is adjusting the filtration. Okay, and the glomerulus is also regulating the composition. That means composition means how much what is the amount of water, electrolyte, nutrients, waste material, that is known as composition of filtrate. So this glomerulus is adjusting the filtration and it is also regulating the composition of filtrate. Then in this tubuloglomerular feedback mechanism, this mechanism stabilizes the nephron performance, and this mechanism is also compensating for fluctuation in blood pressure. So even if the blood pressure is fluctuating, if the blood pressure is high or low, in that condition also the GFR is maintained, it is regulated, and the filtrate composition is also regulated by this tubuloglomerular feedback mechanism. Now in this tubuloglomerular feedback mechanism, a structure which is involved, that structure is known as juxtaglomerular apparatus. And where this juxtaglomerular apparatus is present, so it is present at the very end of the nephron loop. Okay, it is present at the end of nephron loop also known as loop of Henle, where it has just re-entered the renal cortex. Okay, and here the nephron loop contact the nephron loop is in the contact is in a contact with afferent and efferent arterioles. Okay, at the vascular pole of the renal corpuscle. So in the figure I am showing you juxtaglomerular apparatus. For example, over here you see this one is the afferent arteriole, this is the efferent arteriole, this one is the nephron loop, and this nephron loop entered into cortex. So this portion, okay, Here the juxtaglomerular apparatus is present. Okay, over here the juxtaglomerular apparatus is present. And vascular pole. Vascular means the pole or the end of renal corpuscle. The it is the end of renal corpuscle where the efferent arteriole are present. Okay, and the end of the nephron loop is present. That side is known as vascular pole. Now we are discussing the structure of a glomerular apparatus. Now see this portion, they are zooming, and now they are showing us. See, this is the juxtaglomerular apparatus. Now see here the nephron loop is shown. Then here you can see the afferent arteriole is shown. Then you see in the wall of afferent arteriole smooth muscle cell they are showing. Then in the nephron loop in the wall on one side, as you can see, the macula densa cells are present in nephron loop. Okay, then you see the mesangial cell. Okay, so all the names. Then here they are showing the sympathetic nerve fiber. Okay, so this is the entire juxtaglomerular apparatus. Okay, now we are discussing the tubuloglomerular feedback mechanism. See, tubuloglomerular feedback mechanism begins with the macula densa cell. Okay, once again I am showing you macula densa cell. Here you can see this is the nephron loop. This is the very end of nephron loop, this one, and inside you can see these are the macula densa cell they are showing us. So this macula densa cell are a patch of slender, they are closely packed cell, they are the sensory cell, and they are present on one side of the nephron loop. Now what happens when the GFR, glomerular filtration rate, is very high? That time you can understand large amount of glomerular filtrate is going to produce. Now this filtrate, first it will enter into PCT, then it will enter into the nephron loop. So as the GFR is high, large amount of filtrate is produced. So in the nephron loop, the amount of sodium chloride, that means the NaCl level, NaCl sodium chloride level is very high in the nephron loop. Okay, so in short, in the nephron loop, NaCl level is sharply elevated when GFR is high. Now what happened? Now the cells of macula densa, they absorb Na plus, K plus, Cl minus, and these ions, this electrolyte, they are followed by water. So water is absorbed by osmosis process. Okay, so let me show you this. See over here, you can understand the high GFR is high, that means large amount of filtrate is form, and this filtrate enters into the, this is the PCT. After PCT, it will enter into the loop of Henle, nephron loop. So in this filtrate, Na plus is there, Cl minus is there, okay, K plus is there, H2O water is there. Now this macula densa cell, they are going to absorb Na plus, Cl minus, K plus. So all this electrolyte, all this ion, once they enter into macula densa, so water also entering into this cell macula densa. So now this macula densa cell, due to the entry of the ions and water, the cell of macula densa, they swell up, they are swelling, okay, their size increases, okay, and as the cell, they, as the macula densa cell, they are swell up. Now this macula densa cell, they secrete ATP from their basal surface. So macula densa cell releasing ATP from their basal surface. Now now one more type of cell known as mesangial cell, okay, the mesangial cell receives or accepts this ATP which are secreted by macula densa cell. So now ATP enters into the mesangial cell, okay. This mesangial cell inside, they metabolize ATP and they convert ATP into adenosine, okay. So inside the mesangial cell, what happens? ATP is converted into ATP is converted into adenosine. Now where this mesangial cells are present? So mesangial cell, they pack the spaces between the arterioles and within the glomerulus. I am showing you in the figure. For example, in this figure, this is the afferent arteriole, this one is the efferent arteriole. So between afferent and efferent arteriole, this purple color cell, this portion are the mesangial cell, and here also these are the glomerulus, blood capillary they are showing, and in between this capillary you can see this purple color cell are the mesangial cell. So what role these mesangial cells are playing? They are receiving the ATP which are secreted by macula densa cell, and they are metabolizing the ATP and converting them into the adenosine, okay. Now this adenosine produced by mesangial cell, this adenosine act as a paracrine messenger, and now this adenosine stimulate granular cell. Granular cell also known as juxtaglomerular cell. So in the figure I am showing you here these are the granular cell, okay, these are the granular cell. Now what are this granular cell? See, granular cell or the juxtaglomerular cells are modified smooth muscle cell, and as we have seen in the figure, they are wrapped around the afferent arteriole, and to a lesser extent they are also present on the efferent arteriole, okay. So on both the afferent and efferent arteriole surface, this juxtaglomerular cells are present. Now this cell, they respond very quickly to this rising adenosine level, okay, and once they are activated by, once they are stimulated by this rising adenosine level, this granular cell, they constrict the afferent arteriole. So now, as you can understand, the afferent arteriole is constricted. That means less amount of blood is entering into the glomerulus, and now less amount of glomerular filtrate is going to produce. And this is how this cell, they are responsible for the reduction of blood flow into the glomerulus by contracting the afferent arteriole, this granular cell, juxtaglomerular cell. And now what happened? Due to this, there is a reduction in GFR, glomerular filtration rate is reduced, and now the negative feedback loop is completed. So in the figure I will show you this negative feedback loop. Okay, before that, see, the mesangial cells of the glomerulus, mesangial cell we have seen in between the glomerulus, these cells are present. This mesangial cell, they can also contract. See, mesangial cell of the glomerulus may also contract, and they are constricting the glomerular capillaries. Okay, so mesangial cell are contracting, and they are also contracting the glomerular capillaries. And you can understand that as the glomerular capillaries are constricted, that means less glomerular filtrate is going to ooze out from blood plasma. Okay, and this is how the filtration is reduced. Now one more function, mesangial cell also form a supportive matrix. Okay, they provide support for glomerulus, they are present in between the glomerulus, and mesangial cell are also involved in phagocytosis process. So by phagocytosis process, they remove the tissue, any tissue debris. Okay, any damaged cell, they are going to remove by phagocytosis process this mesangial cell. So these are the function of mesangial cell. Now just beyond the juxtaglomerular apparatus, a DCT is present, distal convoluted tubule is present. DCT has a limited capacity for NaCl reabsorption. DCT cannot reabsorb large amount of NaCl. Now this feedback mechanism that we have studied, tubuloglomerular feedback mechanism process, this process help to prevent overloading of the DCT with NaCl. So this tubuloglomerular feedback mechanism reduces the load of DCT to reabsorb the NaCl. Okay, and thus prevent excessive NaCl and water loss in the urine. Okay, so that means all the NaCl, most of the NaCl and water is reabsorbed by this tubuloglomerular feedback mechanism, and this mechanism also reduces the workload of DCT. Okay, and we are not excreting NaCl and water in large amount in our urine. Okay, now here the loop, the negative the loop, okay, that we have discussed throughout this lecture. So in the figure they are showing this tubuloglomerular feedback mechanism loop. So first of all, see, when the GFR, glomerular filtration rate, is high, that time what is going to happen? So due to high GFR, large amount, large volume of glomerular filtrate is produced. Now increase NaCl load in nephron loop. NaCl is present in large amount in nephron loop in glomerular filtrate. Now the macula densa of nephron loop, this cell, they swell up, okay, they secrete ATP. Now this ATP, they are received by mesangial cell. Mesangial cell, they metabolize the ATP and they convert ATP into adenosine. Now this adenosine act as a paracrine messenger, and mesangial cell stimulate the granular cell. Okay. Now afferent arteriole constrict. Okay. So is as the afferent arteriole is constricted, what happened? Less amount of blood is going to enter into the glomerulus. So now less amount of filtrate is going to produce. That means the GFR is reduced. Now glomerular filtration rate is reduced. So this one is the tubuloglomerular feedback mechanism loop. Okay. Now one more, see, sympathetic control. Here in the juxtaglomerular apparatus, here they are showing this is the sympathetic fiber. Okay, this one is the sympathetic fiber. So now we are discussing the sympathetic control. What is the sympathetic control? See, sympathetic nerve fiber richly innervate the renal blood vessel. Okay, sympathetic nerve fiber, they innervate the renal blood vessels. In the figure, as you can see, the fiber has innervated the afferent arteriole. In strenuous exercise, when a person is performing heavy exercise or vigorous exercise, that time, or in some severe condition, drastic condition, or in under the acute condition, for example, the circulatory shock, person when experience, person when suffering from the heart attack, a sudden heart attack, then sympathetic stimulation. Okay, that means sympathetic stimulation means fight or flight condition, and adrenal epinephrine. That time, in this situation, adrenal epinephrine constrict the afferent arteriole. We know, we know the we have in the previous lecture we have discussed the sympathetic preganglionic fiber innervate the adrenal medulla. In the adrenal medulla, chromaffin cells are present, and the chromaffin cell, they produce the epinephrine and norepinephrine. But here we are discussing the epinephrine. So this epinephrine hormone entering into the blood, and then finally epinephrine is going to reach at the level of afferent arteriole, and here this epinephrine is constricting the afferent arteriole. So diameter of the afferent arteriole decreases. Now as the afferent arteriole is constricted, diameter is small, so due to that the glomerulus will receive less amount of blood, and this will reduce the GFR and urine output. Okay, so now under this drastic conditions, uh, uh, kidney will be supplied with less amount of blood, because other body organ, for example heart, brain, skeletal muscle, will require large volume of blood. Because let's say in a fight or flight situation, the person is fighting, that time skeletal muscle due to the cellular respiration in the muscle cell. More oxygen is required, more cellular respiration is going on, more ATP is produced. So that's why the muscles require a large volume of blood. Okay, so in this drastic situation, kidney will be deprived from blood, and the blood will supply more to heart, brain, and skeletal muscle. So under such condition, GFR may be as low as few milliliter per minute. Normally, we know that it is 125 ml per minute. Okay, but in this drastic condition, GFR may be as low as few milliliter only. Okay, so this is about the sympathetic sympathetic control. Now I would like to show you some figures. See here, over here in this figure, the juxtaglomerular apparatus is shown. Over here in this figure also, juxtaglomerular apparatus is shown. And this is the histology slide. Kidney cortex, juxtaglomerular apparatus, stained with hematoxylin and eosin, and we are observing under medium magnification of the microscope. You read all the names. Over here they are shown: distal convoluted tubule, see the macula densa cell, then you see the juxtaglomerular cell, then glomerular arteriole they are showing, then glomerular capillaries, distal convoluted tubule, and over here proximal convoluted tubule, glomerular capsule, two layer, parietal layer, visceral layer, urinary pole. Then between the parietal and visceral layer, capsular space is present. Collecting tubule they are showing, basement membrane is there. So this is the cross section through juxtaglomerular apparatus. This is also histology slide showing the juxtaglomerular apparatus, kidney cortex, renal corpuscle. So you see the glomerular capsule, two layer, parietal, visceral layer, juxtaglomerular apparatus they are showing, juxtaglomerular cell, macula densa, podocyte are shown, capsular space you see, distal convoluted tubule, podocyte, proximal convoluted tubule, distal convoluted tubule. Now here you can see, okay, macula densa, then the vascular pole where the afferent and efferent arteriole, urinary pole from where the proximal PCT, proximal convoluted tubule is arising, that is urinary pole. Okay. So here in this figure, all the names, structure of the renal corpuscle, juxtaglomerular complex, so with this we have completed this lecture on regulation of glomerular filtration. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.